Welcome to our lecture on the fundamentals of biomolecules. Today, we are going to explore a crucial topic in biochemistry known as enzyme cofactors. If you have ever wondered how our bodies actually use the vitamins and minerals we eat every day, you are in the right place. We will build our understanding from the ground up, so do not worry if this is completely new to you. Let us dive in and discover what makes these biological machines tick. To start, let us think about a concept we are all familiar with. Imagine buying a brand new, top-of-the-line flashlight. It has a perfect metal casing and a brilliant bulb. But if you try to turn it on without putting batteries inside, it simply will not work. In the biological world, many of our enzymes are exactly like that empty flashlight. They are perfectly built protein machines, but they need a helper molecule to actually perform their chemical jobs. These essential biological batteries are what we call enzyme cofactors. Without them, the enzyme is just an inactive shell, waiting for the power to turn on. Now that we have our flashlight analogy in mind, let us look at the formal biochemical definitions. An enzyme cofactor is defined as a non-protein chemical compound, or sometimes a metallic ion, that is strictly required for an enzyme to function. When the enzyme is missing this cofactor, it is inactive, and we call this empty protein shell an apoenzyme. However, when the cofactor successfully binds to the apoenzyme, it forms a complete, fully active unit. We call this fully assembled working machine a holoenzyme. Understanding the difference between these two states is the first major step in mastering enzyme kinetics. Building on the idea of the holoenzyme, it is important to know that not all biological batteries are the same. Cofactors generally fall into two main categories. First, we have inorganic ions, which are simple metals like magnesium, zinc, or iron. Second, we have complex organic molecules called coenzymes. Interestingly, these coenzymes are very often derived from the vitamins in our diet. Furthermore, there's a special subcategory. If a coenzyme is tightly or covalently bound to the enzyme on a permanent basis, we give it a specific name. We call it a prosthetic group. Each type plays a unique role in cellular chemistry. Understanding these categories leads us to a very important question. Why do proteins need these helpers in the first place? The answer lies in chemical limitations. The amino acids that make up proteins are fantastic at certain tasks, like acid-base catalysis, but they are terrible at carrying or transferring electrons. Cofactors step in to provide these missing chemical abilities, which is crucial for processes like generating cellular energy. In the real world, this is exactly why humans must consume vitamins and minerals. If your diet lacks these precursors, your enzymes lack their cofactors, leading to severe diseases like scurvy or pellagra, simply because your biological machines cannot turn on. Before we move on to the mathematics of how this works, let us visualize this assembly process. On the screen, you can see a clear schematic of what we have been discussing. On the left is the apoenzyme, shown in gray. Notice its empty binding pocket. It is completely inactive. Next to it is the blue cofactor. Following the arrow to the right, you see the holoenzyme. The cofactor has slotted perfectly into the apoenzyme, changing its state to active. Because the cofactor is now in place, the enzyme is finally ready to accept the substrate, shown at the bottom, and perform its chemical reaction. This physical binding is the core of enzyme activation. Now that we have a clear visual of the binding process, we need a way to measure exactly how strong that connection is. Biochemists use a mathematical concept called the dissociation constant, represented by K sub d. This formula mathematically describes the balance between the broken apart pieces and the fully assembled machine. In this equation, K sub d is the dissociation constant, measured in molarity. The term E in brackets stands for the concentration of the free, inactive apoenzyme. The term C in brackets is the concentration of the free cofactor. Finally, EC in brackets represents the active holoenzyme complex. If the value of K sub d is very small, it means the denominator is large, indicating that the cofactor binds very tightly. Conversely, a large K sub d means weak binding. Let us put this formula into practice with a concrete example. Suppose we are in a laboratory studying a metabolic enzyme. We're given the free apoenzyme concentration, the free cofactor concentration, and the dissociation constant. We are asked to find the concentration of the active holoenzyme complex. To solve this, we start with our standard dissociation constant formula. Because we want to find the bound complex, we algebraically rearrange the equation to isolate EC on the left side. Next, we simply plug in the numbers provided in the problem. The 10 to the minus 6 terms cancel out beautifully, leaving us with our final answer. Physically, this result tells us exactly how much of the enzyme has successfully recruited its battery and is ready to catalyze reactions under these specific cellular conditions. Having successfully calculated the active concentration in a random scenario, let us look at a very specific and important condition. In biochemistry, researchers frequently need to know the exact point at which exactly half of the total enzyme molecules in a solution are activated. This 50% activation point is a crucial metric for understanding how sensitive an enzyme is to changes in cellular nutrient levels. For exactly half the enzymes to be active, the concentration of the free, empty apoenzyme must exactly equal the concentration of the fully bound holoenzyme complex. Mathematically, we write this condition as the concentration of E equals the concentration of EC. This simple equality is incredibly powerful. Let us see how this 50% condition plays out in a real calculation. Imagine a researcher wants exactly half of their enzyme molecules to be active. They know the dissociation constant is 3.5 times 10 to the negative 7 molar. They need to find the exact concentration of free cofactor to add to the test tube. We start by stating our 50% condition, where E equals EC. Then we take our standard equilibrium equation. Because E and EC are equal, we can substitute E into the denominator. The E terms in the numerator and denominator perfectly cancel each other out. This leaves us with the free cofactor concentration exactly equal to the dissociation constant. So the required concentration is simply 3.5 times 10 to the negative 7 molar. The calculation we just performed reveals something much deeper than a single answer. It hints at a universal rule. Let us formally derive this rule to understand exactly why it works every single time. Our goal in this first part of the derivation is to set up the mathematical foundation. We always begin with the fundamental equilibrium equation, which perfectly balances the free components on top with the bound complex on the bottom. Next, we state our logical condition. If exactly half the enzyme is bound and half is free, then the concentration of E must equal the concentration of EC. This setup is the key to simplifying the complex fraction in the next step. Now that we have our setup, let us complete the derivation. Because the values of E and EC are exactly equal in this scenario, we can substitute E for EC in the denominator of our fraction. Once we do this, the mathematical path becomes very clear. The E terms in the numerator and denominator perfectly cancel each other out. This mathematical cancellation leaves only the term C on the right side of the equation. We have beautifully simplified a complex relationship into a direct equality. We have just proven a fundamental biochemical rule of thumb. Exactly 50% of the enzyme molecules will be activated when the free cofactor concentration exactly equals the dissociation constant. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember the flashlight analogy. Cofactors are the essential non-protein batteries that turn inactive apoenzymes into fully functional holoenzymes. Second, these helpers provide chemical abilities that proteins lack, which is exactly why vitamins and minerals are vital for human health. Finally, we learned that we can measure this binding affinity using the dissociation constant. And through our derivation, we proved that an enzyme reaches exactly 50% activation when the available cofactor concentration matches that constant. You now have a solid, fundamental understanding of enzyme cofactors and how they operate.